When I recently was talking to a colleague of mine about this topic, he said that biochar, that's just another form of soil organic carbon. And if you think that way about biochar, then I hope to convince you that it's quite different. Now, to look at our study, we are working in the area that is in the northeast of the Athabasca oil sands deposit, and this is actually the area that is susceptible for surface mining. And to reach the ore body, all overburden needs to be removed. And of course, that means that we turn pristine boreal forest into a landscape that is not that friendly for living organisms. And in Alberta, operators are mandated to reclaim to equivalent capability that is regulated in the Conservation and Reclamation Regulation and in the Public Lands Administration Regulation. The target for reclamation in an upland forest environment are type A and B ecosites, and the soils we find in those ecosites are soils of the Brunicolic order. Those are characterized by a high mineral component, only a very shallow AH horizon, if there is one at all. Another component of these soils is biochar. As you can see in the picture to your left, there is some char in the soil. And I think, meanwhile, you have heard the talk of Dr. Stewart and also Dr. McKenzie yesterday. So, you should be familiar with the positive effects of biochar by now, at least I hope so. And, the dominating tree species on those sites are jack pine and aspen. And if we look at those sites in the reclamation environment, for upland reclamation, we're using peat mineral mix, or in this case, it's more of a straight peat, which you can see in the picture to your left, or forest floor mineral mix, which you can see in the picture to your right. So those sites are both same age and probably 50 meters apart. So there's an obvious difference, and I would suggest that the trees on the peat are not doing very well. So why do we have to use the peat? 70% of the land before disturbance were bogs and fens, and 30% were upland forest. So now we have to use the peat out of the bogs and fens for reclamation. And peatlands will only be 30% after mining, while we have 70% of upland forest. So for salvaging the material, we're using the top 1 to 3 meters of the peat, while when we're using forest floor mineral mix, we only salvage the top 15 centimeters. So that implies that there is a difference in the ratio of material that is available. So now to look at our hypothesis. As the ecosites are having a high mineral component, we were suggesting that by adding mineral subsoil to the peat, we could improve the growth of aspen in our trial. That's the black curve you can see. And we were suggesting that if you're adding biochar, we see a positive effect as well. And we were hoping that we can improve the characteristics of peat that much that it is as good as forest floor mineral mix. So to test that, we set up a greenhouse study with the following experimental design. We had straight peat, P100, and then different mixtures till S100, what is straight subsoil. And as reference, we had peat mineral mix 2, which is a peat mineral mix that performs very well in the field, and we had forest floor mineral mix, which also performs very well as reference substrates. And of course, we had a biochar amendment of each treatment. We use 10 tons per hectare, and that's a fairly high rate, but we decided to go with 10 tons per hectare as we wanted to see an effect in a greenhouse study that was only lasting for 3 months. So we set up the study with a completely randomized design in the University of Alberta greenhouse, and we established a controlled watering regime. We were watering daily, and we were keeping the soils at field capacity, or about 80% field capacity. So then after the study, or during the study, we measured soil available nutrients with PRS probes. Then after the study, we measured biometric parameters, and following analyzed our leaf nutrient contents with ICP-OES. So let's look at the biometric data. What you can see here are our different treatments. Forest floor mineral mix to your right, and forest floor mineral mix was performing very well. But just by admixing mineral subsoil, we could increase the performance of aspen growth regarding the accumulation of biomass that much that there was no more significant difference in between those two treatments. So now, what was that related to? We looked at the nutrients which you can see here. This is a principal component analysis. We have principal component 1 at the bottom, which explains here 60%, and then principal component 2 on the y-axis, which explains 20%. And as you can see, by admixing subsoil, we have a shift of the nutrient profile to the right, so that we can say that the content of magnesium, calcium, nitrate, and sulfur are similar to forest floor mineral mix. The difference in between those substrates that we still see here is due to a difference in potassium and phosphorus content. So if we would apply those nutrients as fertilizer, we would get a really similar nutrient profile. If we look at the micronutrients, there's a lot of overlap. So I only want you to focus on P50, S50, and forest floor mineral mix, which you can see here. This is P50, S50, and forest floor mineral mix. And also the micronutrient profile is very similar. There is a difference in manganese and lead, but it's very similar. So now, when we look at the effect of biochar amendment, we can see that green is with biochar, red without, that by admixing or amending biochar, we get a positive effect on growth in most treatments. You might say that's not significant, that's described for biochar, but we have one treatment, peat mineral mix 2, where the effect was highly significant. And so we can say that the effect of biochar is quite substrate specific, but in general it's positive, but we can also have a slightly negative effect, as you can see here in the subsoil. So let's talk about the positive effect in peat mineral mix 2. So we were looking at the nutrient profile again for principal component analysis, and using the PRS probe data. And as you can see, there's an overlap going on. So there is not quite a significant difference in between those treatments in the macronutrients and also in the micronutrients, which you can see here. We have that overlap. So for this treatment, I looked at the leaf nutrition, and what we found here is the following. Here we get a quite significant difference in between the treatments. That's probably due to sodium. And sodium is not measurable with PRS probes. I actually called Eric and asked him for sodium data, and he said, oh, we don't have your samples anymore. Unfortunately, I was a bit too late. And yeah, so here the difference is probably due to an absorption of sodium by biochar. And as we know, the ratio of sodium to potassium in the plant leaf or in the plant is an important factor for growth, right? So here, that is probably what causing the significant difference in growth of aspen. So now to conclude our findings, we can say that mixing mineral subsoil and peat can lead to substrate quality that supports growth of aspen comparable to reference substrates for mineral mix. Peat biochar improves biomass production of aspen on most substrates, but the effect is substrate specific. And the amendment can lead to a highly significant improved productivity in substrates, here especially in peat mineral mix 2. And the biochar effect on aspen growth is related to changes in the nutrient profile. Finally, I would like to acknowledge our helpers and research assistants, Jennifer McInnes, Cillian Fu, Monica Schandau, Patrick Newberger, William Kirby. Especially Jennifer was a great help. She's here today as well. She was really a good help in the greenhouse study and with the field work. And I also would like to acknowledge our funding organizations, the University of Alberta, Department of Renewable Resources, Include Canada, and Circulate, and the Land Reclamation International Graduate Student.